Hello and welcome to the Medicine and Machine Learning Podcast. I'm your host, David J.H. Wu, and in today's episode, I interview two very special guests from Relay Therapeutics in Cambridge, Massachusetts. My guests are Pat Walters, the Chief Data Officer, and Patrick Riley, the Senior VP of AI. We talk about Relay's innovative approach to drug discovery and how new developments in AI and computational modeling have really accelerated this process. This was a super cool interview and has lots of gems for anyone interested in how we discover the newest drugs in medicine. So I hope you all enjoy. Thank you. So hello, everyone. Welcome to a new episode of the Medicine and Machine Learning Podcast. I'm your host, David Wu, and today I have Pat Walters and Patrick Riley from the computational group at Relay Therapeutics in Cambridge, Massachusetts. So for our first question that we ask every guest, I was wondering if you could tell us about your path and how you came to the intersection of medicine and machine learning. And I was thinking maybe Pat, starting with you.
Sure. Yeah. My scientific career, I started as an organic chemist and I was working at the bench and fell into computation more out of laziness. There were aspects of my experimental work that I found tedious. So I decided to start working on automating some of those. After that, decided that I did have a bit of an aptitude for computers. I really liked doing that sort of thing. Ended up going to graduate school and studying applications of artificial intelligence in conformational analysis. After finishing my PhD, I went to work at Vertex Pharmaceuticals in Cambridge, Massachusetts. I was there for 20 years and eventually ended up being the global head of modeling and informatics, and really started from the time I started working there in the mid-1990s, started working on a variety of applications of machine learning on drug discovery problems. Moved to Relay Therapeutics about 6 years ago, started the computational group, and then continued to work. And then about a year ago, Relay acquired a company called Zebi AI, brought in a larger AI and machine learning group as part of that Patrick heads.
Oh, wow, that's a perfect segue. I was wondering if Patrick, you could kind of tell us as well what brought you here.
Yes, I had much more of a meandering path before I got into the pharmaceutical industry. So I came, my training's all from the computer science side, and even from when I was an undergrad, I was fascinated with this idea that machines could be intelligent and that they could be as intelligent as people, at least in some ways, and that that very quickly became my passion from the computer science side. Went through, got my PhD. And then I went off and spent a long time at Google. I would spend time at Google search, and really, during that process, really kind of rediscovered some of my passion for the really large data problems, statistical analysis, data analysis, and how that all intersects with these questions of how machines can do intelligent things. And so, at that time, Google was really very prominent in this world of big data analysis. I like to tell people I was doing big data before it was cool. But after a while, I really wanted to do something new. I was tired of thinking about the problems inside of Google search, and wanted to really explore some new ones. And at that time, inside of Google Research, there was a team forming called Google Accelerated Science. That was looking at applications of AI, ML, all kinds of computational techniques to a broad range of scientific problems. And so I got exposure and got to work with a lot of great scientists from places as diverse as agriculture and nuclear fusion to material science and chemistry and drug discovery. And through that process, I really had a growing interest in what we could do with chemistry, in particular chemistry for drug development. And I saw real opportunities for how the AI and ML techniques could really have an impact on what was going on and how drugs were being developed. And when the ZBA acquisition was happening, it was really like the stars aligning for the perfect place for me to make the jump from more of a pure research position into a much more applied position here at Relay.
Wow, that's really cool, and I'm glad that I have both of you here today on this podcast, because I feel like, it's a little bit of yin and yang, but similar, but a little bit different, and it'd be really cool to get both of your perspectives.
I think it's part of the, part of why I wanted to come to Relay was to work with Pat because of all the knowledge and expertise that he has, and the team that he's built here is it's really quite an amazing place.
Did you guys know each other beforehand?
We did. We'd actually collaborated when I was at Vertex about 10 years ago. So we were doing some of the first work with TensorFlow, actually before TensorFlow was TensorFlow.
Whoa. What was TensorFlow before it was TensorFlow?
So there was an internal deep learning system that we had at Google before that, and then TensorFlow was being built. And so before it was even released publicly, we Steve Kearns, that I was working with at Google, got to use some of the not yet released methods as part of this collaboration we had between Google and Vertex at the time.
Yeah,
Wow.
and as part of things being a small world, so Steve Kearns now works at Relay Therapeutics, as does Brian Goldman, who was the other person on that project. So we're all kind of back together again.
That's so cool. It's like the Avengers coming back together to tackle. And actually, that kind of takes me to my next question. So, you're both at Relay Therapeutics now, and I was wondering if you could tell us, what does Relay Therapeutics do?
Sure, I can take that one. So Relay Therapeutics is a biotechnology company. We're really a drug discovery and development organization. We started this company 6 years ago with the idea that we could operate at the intersection of computation and experiment. We really believe that in order to do things like build good machine learning models, we really need to have high quality data. So we wanted to create a company where experiment and computation were on an equal footing and use that to push things forward. We have now grown the company to about 300 people. Over the last 6 years, we've been able to put 3 compounds into clinical trials. So right now we have our first compound was RLY 1971, which is now GDC 1971, and that's being tested in a clinical trial for patients with solid tumors with G12C mutation. And that's in collaboration with Genentech. Together with GDC 6036, which is their KRAS G12C inhibitor. Then we have a PI3K, a mutant selective PI3K alpha inhibitor, RLY 2608, and an FGFR2 inhibitor, RLY 4008. So we've been able to progress all of those through the combination of our computational and experimental efforts.
Wow. So I guess briefly for our audience, those are each tumor markers, right? Or the FG or genes targets, right? Or could you kind of really briefly like talk us through?
Yeah. Those are proteins that are mutated in cancer. So what will happen is, for instance, PI3K alpha, you end up with, there are 3 predominant mutant forms of PI3K alpha that are tumor drivers. And when these proteins are mutated, then you end up getting tumor formation.
And how do you guys target those proteins?
Typically, we've been going after targets that have a genetic precedent. So in many cases, people have been able to put compounds into clinical trials that inhibit these targets and have seen reductions in tumor volume in patients. The problem is that the majority of these compounds are not selective. So in addition to hitting the target of interest, they'll also hit closely related proteins and bring about side effects. And because of these side effects, the patients typically have to go off of therapy. So what we've been focusing on is using this combination of computation and experiment to design selective molecules that only inhibit the target of interest. So this really comes about through a combination of biology, biophysics, biochemistry, structural biology, and then we can use all of this experimental data to drive our computational models, form a hypothesis, and then do additional experiments. So the way that we think about things is every model that we build, every calculation that we do is there to design the next experiment.
Wow. So is most of it in silico, or you guys start in silico first with computational models? Or
so typically what we'll do is we'll see a particular protein that we know is a genetic driver. Then what we'll do is we'll computationally and experimentally model that protein. So for instance, with PI3K alpha, our structural biology group was able to obtain the first X-ray crystal structure of the full-length protein. Wow. We can then take that protein once we have it, and we can simulate it using a technique called molecular dynamics. We can simulate the motion of the protein and then try to use our computational methods to develop hypotheses for how we can inhibit the activity of that protein.
Is that oh yeah, please.
I want to emphasize something that Pat's saying there, which is this, so many people talk about this idea of, oh, can we do a purely in silico drug development? And that's just not the way it works. The key thing is this interplay back and forth between computation and experiment. I don't think we're ever going to be at a point where someone just says, I have an idea, I'm going to run the computer and the drug's just going to pop out. That's just not the way things work today, nor do I think it's the way it's probably ever going to work.
That's really good insight. Wow. I'd love to hear more.
Sure. Let's walk through the process.
Kind of just to give the lens of a medical student. We know almost nothing about how these drugs come into play. All we just know is what they target. If we get a little fancy, these are antibodies or monoclonal antibodies, but we really don't know how they come to be about. So, this is really fascinating to hear about.
Okay, so maybe we can walk through the process and then kind of point out some of the places that machine learning and AI are a role. So for instance, let's say we've identified a protein. We have an idea that we feel that we could modulate the activity of that protein, and then we want to find a chemical starting point for this. So we can do that in a variety of ways. One way we could do that is to take that crystal structure of the protein, and we could take billions of small molecules and fit them into that protein's binding site to see which ones fit.
And do that computationally, you're saying?
Yeah, exactly. We can do all of that as a computational exercise. It's a technique that we call docking.
I think I've seen some figures on your guys' website of the, them doing it over and over with the protein and little molecules coming in. Kind of, they're really cool figures. I suggest to any listeners to check out the website because, I guess a figure is worth a couple thousand words, right?
Right, but some of these techniques can be computationally expensive, so it can take a long time to run these calculations. So one of the techniques that we can use to streamline that is a machine learning technique called active learning.
And there, the idea is that rather than having to actually run the computation on all 10 billion, which can be very expensive, you run a computation on a small set. You build a machine learning model that then estimates how all the rest of them would do. And then you kind of do this in a few rounds, so you give the model a chance to learn a little bit more each time. And then you don't have to run all those calculations, but you can still have a pretty good idea, these are the ones that are going to score the best. and that we should consider then taking the next step and saying, are these now worth actually trying in the real world? Because as good as these computational techniques are, they're still just an estimate. They're kind of an educated computational guess of what's actually going to work in the real world. And you don't know until you then actually make those compounds, buy those compounds, and then really go and test them against the actual physical protein.
So just curious, do you guys have like a database of 10 billion small molecules or compounds that you like kind
Yes.
of like that's like the ocean, and then you guys kind of sample in little buckets to like train the machine learning algorithm? Is that kind of like a good analogy?
Yeah, no, that's actually a really good analogy.
Right. But I think this emphasizes one of the things that's really hard about this, design of small molecules. People have tried to estimate the number of small molecules that could be potentially interesting drugs, and the estimates vary quite a bit. But it's from like 10 to the 30th or like 10 to the 60th, right? I mean, these are enormous number of molecules we could make. And so that's one of the things that fundamentally makes this both a really hard and
like more than the number of stars in the universe, right?
Yes, exactly. Absolutely, right? So there's so many things we could possibly try to make. How do we find the one that actually is going to be this selective inhibitor, right? This is, in some ways, this is what the small molecule drug discovery challenge is, right? There's so many possibilities, and we have to navigate that space using our computational tools, using our experimental tools.
Wow. And I think, excuse my basic understanding of proteins, but I'm imagining, for a certain target, is it, do we expect there to exist like a magical molecule or compound that will like fit only the disease active proteins or the cancer causing proteins, and then inactivate them or cause some kind of cascade, and then the other proteins are fine? Does that kind of make any sense? Or
yeah, absolutely. So just perhaps to just take a step back and answer your question. So first off, what we're trying to do is find a starting point. That starting point is not going to be the drug, but it's going to be something that sort of fits in the binding site. And then what we want to do is modify that and continue to optimize it, to make it to make sure that it's going to be bioavailable. And then as we're modifying that molecule, we're also looking at closely related proteins to make sure that it doesn't bind to those.
Wow.
And then we'll also be running what we call panel assays over the course of this, where what you'll do there is once you have a molecule that you think is promising, you can go and run that against a panel of, let's say, 500 different proteins, just to see.
Like naturally occurring human proteins?
Yeah, exactly.
Okay.
Just to see, okay, is this molecule going to be promiscuous or is it going to be selective? But one of the things that we focus on is trying to optimize the selectivity of our molecule. So make sure that the molecule binds to our protein of interest, but doesn't bind to closely related homologous proteins that could cause side effects for patients.
And the important thing to realize is we talk about binding or not binding, but that's actually that's not a binary thing, right? Almost every drug has a little bit of off-target activity somewhere. And it's just a question of it's not enough to cause a clinical problem for a patient. And so it's not, we need to move out of this world of thinking about, oh, it only hits this. Well, it hits that protein much, much better than the one nearby, right? But that doesn't mean you could, if you gave enough of it, it would hit that other protein as well, right? And so it's all kind of a continuous and space that we're dealing with here.
That is so cool. You know what it reminds me of? It reminds me of VDJ recombination in when humans make antibodies. It almost reminds me like it feels like, how like our cells make antibodies, and there's like this like infinite combinations of like, it's kind of how, and then they test those antibodies like on our own cells and then on antigen presenting cells. It feels like we're doing this ourselves, like on a bigger macro level, like using like AI, machine learning, testing 10 to the 30th 10 to the 30th compounds. Instead of doing this in our bodies, like in our thymus, we're doing this like now outside in labs. You know what I mean? Is that is
No,
that a
it's
fair
a,
analogy?
it's a great analogy, right? Because, I would say in some ways the body is doing a very, kind of uneducated exploration, and random exploration, by the way, is a really good strategy a lot of the time. And so, the job that we have here is how do we use our computational techniques, use our AI techniques to do a little bit more of a guided exploration, right? So you don't have to keep making everything, saying, you know what, only make these ones. Right. So rather than having to make as many and your body is basically making and testing all those antibodies, right? Can we make a lot fewer? And, because part of the challenge here is with the way we do things here is we're honestly, our body's really good at making many, many, many different antibodies. Mostly we're not as good at making many, many different kinds of small molecules to test. It's a slower and more expensive process, right? Now, there are ways that we can make very diverse, very large libraries in some cases, and we could talk about those. But I think this idea of how you guide that search using all these computational methods is really fundamental to how we think about the world.
Wow, I think that is a beautiful, ingenious idea.
And so in addition to these computational approaches that I described to finding starting points, we also have experimental approaches. So one of the techniques that we use is a technique called DNA encoded library screening. So there, what we're doing is we're synthesizing chemical libraries of millions or even billions of small molecules. Each of these small molecules then has a DNA tag associated to it. So then you can take those small molecules, incubate them with a protein, and then wash away the rest of the small molecules. The molecules that bind to the protein, then you can decode that DNA tag. Each DNA tag is associated with a specific small molecule.
Oh, wow.
So we can do that, but we can actually go beyond that and use all that data from the millions or even a billion molecules that we screen and use that to build a machine learning model.
And this is one of the things that I had an opportunity to do while I was at Google, and kind of one of the jumping off points for me here to come here was to show that you can build very effective machine learning models on that kind of data. And so that way you can sort of generate lots of data experimentally, make this transition over to the computational world, say, great, I have all this data from the experiment, build a machine learning model. And then from that model, we can go back and say, okay, what do we want to make and test in the real world next? Right. And this is another example of this kind of back and forth process of experiment and computation informing each other.
I feel like this second example was a bit harder for me to grasp. I was wondering if, kind of speaking in analogies, if you guys had like a really good analogy for helping understand it.
I would think about it this way. So, if you want to, test lots and lots of molecules, one of the things you could do is you could, pick one, test it, look at that one result, pick another one, test it, and you're sort of going one at a time, right? The alternative to that is to just imagine you could take something where only the thing you want sticks to it. And then you just like flow all the molecules by. You just, you go to a river, you throw in a fishing pole, only the fish you want will grab your bait, right? Therefore you don't have to test every single fish. You drop your fishing in there, you let all the fish come by, and the one that likes the food you're putting out there grabs onto it, right? And that can be a way that you can effectively test many, many, many small molecules without having to do them all one by one. It's this kind of selection process where you let them all go by, and whatever small molecule likes this protein will stick to it.
So in this case, the bait would be the protein, right? Yep. And then you're running all of these small molecules across it, and then the one that bites is the fish, the prize fish, right? That's like the one that's going to target your protein. And the way that you know which you found your fish is that there's like a DNA tag attached to it or something.
It's just they'll have a little barcode. right?
Yeah.
Okay.
It's like they have little dog tags identifying each one of the fish that are in the
Oh,
that are in the
wow.
pond. So you do that, and then then you say, great. Now, how do I know what's there? Well, go look at everybody's barcodes, right? You can grab all the
Wow.
barcodes, and then you read them out with DNA sequencing. And I think this is actually kind of interesting. This is another example where people think about, oh, the DNA sequencing revolution has changed our ability to do genomic sequencing, understand our genomes. And that's true, but it's also opened up a whole lot of experimental techniques that allow us to interrogate things that are not just our genome, but the DNA sequencing technology that allows you to see your genome also enables this other kind of experiment, right? That has nothing to do with your genome. We just use DNA because it's a convenient way to write a
Wow.
barcode
Okay.
and
Okay.
attach it to
Yeah.
a
Yeah.
small
Yeah.
molecule.
I was just gonna ask that question, but
Yeah.
yeah, thank you for answering that. It's just because it's convenient that we use the DNA.
Yep, because it's a thing we can read a lot of very, very, very efficiently.
Yeah, but I guess building on that analogy, there's a couple ways that you can do this. One way is you would have your fish that grab onto your bait. And that way I do that and 3 fish grab onto my bait, but I didn't learn anything from all the fish that swam by, right? So by building a machine learning model, what I can do is I can say, here are the fish that grabbed the bait. Here's all the fish that swam by. What distinguishes the fish that grabbed the bait from the fish that swam by? Now, can I use that to go and find a whole bunch of other fish?
Wow. Yeah. Wow.
So it's not just trying to use the technique to just find a few hits. It's trying to use the technique to build a model that lets me understand the sorts of molecules that are going to bind.
This reminds me of when you were talking about when you were doing your PhD, you started out in organic chemistry, but then you were tired of doing experiments and you wanted to like computationally save time by doing them. Like, this is like saving a lot of work in a sense, right?
Yeah, exactly.
Yeah, if we do our job right, then we have to run the right experiments, right? And we can do this whole process faster.
Wow. So I think we've talked about 2 strategies that you guys employ right now, right?
Yeah, and there are a number of other strategies we can use to find starting points, but maybe we can segue from there to talking about how we optimize these molecules.
Yeah,
so
let's do it.
We found these starting points, we have them in hand. Now we want to optimize those molecules so that they'll bind to the target that we want to hit, but not bind to a whole bunch of other targets.
That's what I thought we were done after we found the fish that
No.
Yeah, that's just the beginning, right? Exactly.
Because we found a bluefish, and what we really want is a green fish. So we've got to figure out how to turn the bluefish into a green fish.
Okay.
So we have a number of experimental techniques that we use to help us understand how that small molecule is binding. So we have techniques like X-ray crystallography that allow us to get a picture of how that small molecule is binding in the protein. So we're essentially sending an X-ray beam through a sample of the small molecule bound to the protein. Then by looking at the X-ray diffraction pattern, we can come up with a picture of what that small molecule looks like in the protein. And that's the snapshot. What we want to do, though, is we want to look at how that small molecule interacts with the protein in something closer to its native state. Because if you think about proteins in cells, they're moving. So we have a technique we use called molecular dynamics, which simulates the motion of the protein and then the small molecule in the context of the protein.
In the cell.
Not in the cell. This is just the protein we're simulating the motion of the protein typically in water.
Okay, got it.
So then we're simulating the motion of the protein in water. We can look at how the protein moves. We can look at how the protein moves in the context of a small molecule and in the absence of that small molecule, see where that's different. We can also model the motion of the protein that we want to inhibit and all the closely related proteins that we don't want to inhibit. So the proteins that would give us side effects. And just as an example, I told you at the beginning, we have an inhibitor of a protein called FGFR2 in the clinic. And so we want to be able to inhibit FGFR2, but we don't want to inhibit FGFR1, which is a closely related protein which regulates phosphate homeostasis. So, if you have an inhibitor that inhibits both FGFR1 and FGFR2, you may be reducing tumor volume, but you're going to be messing with phosphate levels in the patient, and this can cause all sorts of side effects. So in the FGFR2 program, we did these simulations and were able to observe differences in motion between FGFR1 and FGFR2 and use those as part of the design process for the molecule.
Wow. That's so cool. So they move differently, FGFR1 versus FGFR2?
Yes, absolutely.
Wow. That's one thing we didn't learn in school.
The binding sites of the proteins are very similar, but there are some subtle differences in the way that they move. So now I told you we can make these movies of these proteins moving, right? But let's say that I've got 1,000 molecules that I'm thinking about making. So I can do simulations of all 1,000 of those molecules. I told you at the beginning, I'm a lazy person. I don't want to sit. Do you want to sit there and watch 1,000 movies of small molecules in a protein binding site, right? So this is another place where machine learning comes into play. So what we can do is we can capture different characteristics of those simulations and then use that to build a model.
And there, just like, we had this problem of like, what makes this fish like this bait? Now we have a question of what makes this molecular dynamics trajectory, what indicates this molecule is selective versus this one not being selective. It's the same problem of, can we have the machine learn the pattern that might be otherwise difficult for us to see? Just now we're looking at what's the difference in these movies.
I want to hear more. Keep going.
Yeah, just, I mean, this is where the kind of broadly what we see for most of our machine learning techniques is we're asking them a question of, can you find me the difference between these things, right? Tell me whether this is a movie from a selective compound or a not selective compound. Tell me if this is a small molecule that binds my protein or doesn't bind my protein. A lot of the machine learning we see, we're asking it this kind of question, right? Wow. Can you please tell me whether it goes in this bucket or that bucket, right? And, we talked about, the DNA encoded library screening and figuring out which molecule is the one I want. This is another example saying, well, which of these movies looks like what I want, right? What's the difference between them? What can you see what can the machine see in the motion to enable it to distinguish between the kinds of molecules we want and the kinds of molecules we don't.
Do you guys get a snowball effect as like the more you do, the more data you acquire, the better you understand, where the different kinds of small molecules that you guys have, and does that make future experiments easier?
Absolutely, but I think it's a little more subtle than this in that it's not just that, that data that we made for this program is useful for that. Part of what happens is once we've gone through this and seen this on targets, we know better about how to interpret the data. We know how to make the model the next time. So it's not just that the raw data point is useful, is that we become more expert every time we go through this
human
process.
Learning too, right?
Yeah, absolutely, right.
Machine and human learning.
Yeah, no, and this is where, if we talk about, what is the platform that we talk about here? It's not really a bunch of algorithms that we have sitting out, but that's part of it. It's the expertise about how to use all these things effectively, about how to go back and forth between experiment and computation. Fundamentally, that's really what the platform is these technical things combined with the expertise of the people that we have here to actually use them.
That's a beautiful thing.
Yeah. And I think the other part about this is this often, as we start analyzing this data and looking at these models, it changes the way that we do experiments. So we may want to say, okay, this didn't really give us the readout that we need. Let's modify the way that we're doing this experiment. So it's a constant interplay between computation and experiment. So we have about a dozen people in the group whose role is to work as part of collaborative drug discovery teams. So there will be one or more computational chemists on the team, and they'll work very closely with biologists, biochemists, structural biologists, pharmacokineticists. So it's really a team effort. And then, backing all that up, we have other groups of people who are doing different algorithm development. So making sure that we have the fundamental computational infrastructure we need to do this, and that we have people who know how to use that infrastructure.
I'm curious, how much would you say AI, machine learning has shifted this process? And I guess I'll give some context to the question. I'm curious, like, what did we do before all this? Did we just manually screen compounds, like compound by compound? And has AI brought like a huge paradigm shift to this process, or is it just made things really much faster and easier?
Well, I would say, if you look kind of broadly, the AI is one part of of computational techniques that have a long history of being developed and incorporated into the drug discovery flow. And so drug discovery constantly absorbs new technologies. At one point, people did many things through animal testing. Now we test many things through animals because we have experimental techniques to understand what's going on with proteins. A bunch of the computational techniques that are now commonplace for everybody to do, types of chemical similarity searches and so on. A couple of decades ago, those were the brand new techniques that everybody adopted. And so really the incorporation of a lot of modern AI methods into drug discovery, it's just part of a long trend of adopting new tools, all along the way. And so in some ways, I don't think that you're seeing a sort of a fundamental break from the trajectory that was already happening of bringing in new tools. And as I think some of this discussion has already brought to light, drug discovery is not just one problem, right? It's a very diverse set of problems and expertise that have to come together. And you're seeing AI methods get applied in all the various disciplines and all the various problems that you're seeing around drug discovery. So there's not one weird AI trick that gives you a drug, right? You're seeing bits and pieces of, oh, look, here I'm going to make my hit finding work a little bit better because I'm using machine learning here. I'm going to make my prioritization of the next compound to make a little bit better because I've learned from the molecular dynamics trajectory. And so really, I think what you're seeing is this kind of, gradual improvement across all these different areas rather than like one fell swoop of everything, everything changing all at once.
No silver bullet kind of.
Exactly. Absolutely.
Yeah, I think the other place that I'm seeing a shift is in people starting to think about, how can I do an experiment to improve my model? I think in the past, it was very much, we're going to do experiments the same way that we always have. We're going to give you computational folks all this data and you can build a model. And, in the past, we would look at that data and say, but that's not the data that we wanted. And I think now there's been a bit of a shift where we're really looking at this and saying, okay, here are the areas where our model is performing well. Here's the area where our model is not performing well. Let's do some more experiments to try to improve the performance of the model.
I mean, so it's more, there's more feedback, kind of like a
exactly.
interplay. Wow. This is really fascinating stuff. I'm so feel honored to be talking to you both right now about this.
Well, we think this is fascinating stuff. That's why we're here.
But if you think about it, it's really it all comes down to, I have some data and I have some labels. And I just want to find the association between that data and that label. So, let's say that I've got a set of images of cells, and I want to be able to have my algorithm learn, which of those cells are cancerous and which are not. So what I can do then is I can train my machine learning algorithm. Here's a set of, here's my set of images. I've got them labeled now as cancerous, not cancerous, and my algorithm is learning the relationship. It's the same thing. I've got this molecular dynamics simulation that I did. I've got a set of movies of those simulations. Can you learn the relationship between that set of movies, which is my data, and the labels, which is selective versus non-selective? So it's always just taking, it's really almost like linear regression. You've got a set of X variables, and you've got a Y variable, and you want to find the relationship between X and Y.
And I guess my next question would be, we were talking about like the trajectory of drug development and kind of how it's been absorbing these new technological breakthroughs from kind of various fields. I'm curious this is also, questions that we ask every guest. What do you think? And normally we ask, what do you think the future of AI in medicine will look like? But for you both, I'd like to know, what do you think the future in drug development looks like in the next 10 to 20 years?
I mean, I think broadly what we're going to see is it's going to become just the thing you do. Of course you do, right? It's right now, I feel like there's a lot of like, kind of attention and like, ooh, what are you doing with AI? Ooh, and I think we're going to get to this point where it's kind of like our cell phones are today. Of course you have your phone with you all the time. Of course you use that AI method. We're not like, talking about it. It's not going to, it's going to be a little less like novel, because it'll be the thing that everyone is just doing, because it'll be like
3G and 4G, right?
Yeah, exactly. It's like, of course you use that, right? And it's going to become kind of, in a lot of ways, I think some of it's going to be unremarkable, because there are some of the things that are going on that are just going to work and everybody's going to do.
Yeah, I agree. And the way that I look at this, there's three parts to building a machine learning model. There's the data, there's the algorithm that you're using. There's the data, there's how you're representing that data in a form that you can use to build a model. And then there's the algorithm that you're using to process that. And I think we're going to see improvements in all three of those areas, but I think it's all going to be driven by the data. The data, to me, is the most important part of this. And as we are able to improve our ability to collect data, as we get to better experiments, we're going to see concurrent improvements in our machine learning models. Yeah, I mean, think about this from the standpoint of genomics, right? We've sequenced lots and lots of genomes, but we don't have the relationships yet between those genomes and some phenotype, some disease state. So, as we start to collect more of this data, I think we'll start to understand more complex biology. I think as we continue to improve our ability to characterize small molecules and understand the interactions between small molecules and proteins, that'll improve our ability to build a model.
So I have some questions that I'd like to ask that are a bit deeper, but more so just for fun. Questions that I'm just kind of curious about. And my first question, it's kind of silly, but it's what do you think a protein actually is? Like, how do you view a protein in your mind? And what I mean by that is I find it so mind-blowing that in our cells, we have like thousands and thousands of proteins that kind of swim around, and each one does like a special thing. And it happens so fast too. And like on the scale is so small, and it's like, it really, I don't know. I almost find it like impossible that how does this all happen so fast? And there's so many like thousands and thousands of little proteins that have special roles and they just happen to find the right things. I don't know. Like, I guess my question is like, what is a protein?
So I think the way I think about this is we tend to, draw a picture of a protein, or we look at a sequence, or we watch a little movie. These are all very simple representations that hide so much underlying complexity of what the proteins are actually doing. But as you're referring to, like, there's so much going on. There's so many complex things that are happening about how they interact. And in some ways, there's no way we can like understand all of that all at one view. And so when I think about proteins and protein targets, we're always thinking about, okay, let me remove a bunch of the things that's going on. What's the simple view I'm going to look at now? Let's look at this crystal structure, or let's talk about this protein as part of an underlying biological cycle. And so I guess in some ways that maybe I'm being defeatist here in some way, but I don't think we can actually like fit in our brains the full set of complexity of what's going on there at any time. We always have to kind of say, okay, I'm going to look at this simple view of it right now. And so in some ways, I think it's all of the things we think of, but even more than that.
Yeah, I remember talking to a cell biologist early in my career who said, you're thinking about this protein as if it's just a ball floating in a swimming pool. And it's not. It's in there, it's embedded in a kelp forest, right? There are just so many other proteins in there. Everything's packed in together. And my hope is that structural biology is evolving at an incredibly rapid pace. New techniques are coming online all the time. And, so my hope is that as the field progresses, we won't just be, modeling a single protein, and we'll start being better at being able to actually model an ensemble state. So several proteins together, and hopefully get to the point where, years from now, we can start modeling how everything exists together in a cell. But it's amazing how far we've been able to get with relatively simple models of a protein in a box of water.
And I guess adding on to that question, I think of the human body, and I feel like at a cellular level, everything kind of makes sense as it like, we kind of, it seems more mechanical, like muscle cells, they just kind of are like pulleys, skin cells are like little barriers, neurons are kind of like little input output devices. But I don't know. I think for me, I just find it almost, it's awesome in the literal sense of like, why is it that when we go down from like the cell level to the protein level, everything just becomes, it seems so much more complex, like you're like orders of magnitude more complex. And I don't know. I guess I'm having trouble like wrapping my mind around like, whoa, I don't know, like kind of like on a deeper poetic level of understanding, like what is, why is the protein world so complicated?
Yeah, well, I think, one of the things that we try to do, and I think, everyone in the field tries to do, is decompose things into model systems. So, you can run an assay with a protein in an isolated environment where it's not in the cell. And then if you then take the same compound, you run the assay in a cell and you see differences, you try to understand what's going on there and why you're seeing the difference. So, is that difference because of the inability of that small molecule to navigate the cell membrane and get into the cell? Is it because the environment that the cell is in is different in the cellular context than it is in when you're just running it as the isolated enzyme? And that's why I think it's essential. And I don't think that's the sort of thing we're ever going to get at through computation alone. And it's really important to be able to say, okay, I have this disconnect. What experiment can I run to help me to dissect this and debug it? And then, based on that, okay, now I've got this experimental data. How can I adjust my model? So, for us, it's really this constant interplay between experiment and computation. And we just to belabor the point, we've really tried to integrate that into the culture of Relay since day one. So, when we started the company, one of the things we did was just, rather than saying, okay, all the chemists go sit over here, all the biologists go sit over here, we mixed things up. And it was great because we would have, chemists sitting next to a biologist or sitting next to a computational person and saying, oh, you're running that simulation. What does that mean? And I think just, having this interplay and having people who are very curious about other disciplines is essential.
And this, I've always appreciated the ability, both in what I've done and people I've worked with, to cross those boundaries. I think that kind of, when you can, I'm never going to be a biologist, but I can learn enough to be able to have a deeper interaction with the biologists about what they're seeing and what they're thinking about. And I ask the same thing, see other people, the biologists say, okay, what is that model actually doing? Like, why does it work?
Yeah.
Right. People, I get asked these questions by other folks at Relay. And I think that curiosity is just is a really important part of having a really kind of vibrant and very productive culture here.
I'm curious for both of you, what is one of your favorite topics in physics, chemistry, or biology? Just like one topic.
I still, I have a lot of my academic training was in organic synthesis, and I still love to geek out and look at elegant routes to complex molecules.
So I will go further down than this. One of the things I spent some time learning about when I was at Google was about the kind of fundamentals of quantum mechanics. Mm-hmm. And I think it is absolutely beautiful. And part of what's going on there is that, quantum mechanics seems so amazingly complicated, and yet you can express everything in a tiny set of rules. And all the complexity is this emergent phenomena from these tiny set of rules of quantum mechanics. And in my mind, that is such an amazing hallmark of what intelligence is, that we're able to simplify down these amazingly complex phenomena that we see from atoms into a tiny set of rules. And that is something that I would love to see artificial intelligence systems do a better job of, is to come up with very, very simple explanations to very complex phenomena. Right now, most of our AI systems are coming up with very complex explanations of complex phenomena, but I think that we aren't going to have true intelligence until we do better at coming up with simple explanations.
Wow. I really like what you're saying, especially because I feel like nowadays it seems like with neural networks, people love to say how they're not explainable. The models that they come up with are kind of like a little black box. But I think, are you kind of implying that maybe one day this black box will become kind of very elegant and simple and almost like kind of meta, like kind of beyond human, our current mortal comprehension kind of?
Possibly. I don't think we're there yet, but that we could certainly end up in that world, but that's not our problem today.
This is one of our closing questions that we ask every guest, and it is, what advice would you give yourself when you were finishing up grad school?
Learn more biology. I think ultimately, it comes out to being scientifically well-rounded and understanding the entirety of the problems. So I wish I had learned more biology. I've kind of learned on the fly, but there's still huge holes in my biology knowledge.
And this is maybe I'll riff off of that, which is just this, you are not going to know all the things you need to know when when you're leaving grad school, and you have to adopt this attitude of, what am I going to be learning next?
Well, Pat and Patrick, thank you so much for this interview today. It was really a phenomenal, inspiring interview. I think Relay Therapeutics is such a cool company, and it's really cool to see how interdisciplinary you guys are, and not only interdisciplinary, but also, synthesizing and synergizing all of the newest technological breakthroughs in AI. Wow.
Well, thank you, David. It's been very great talking to you today.